Welcome students back to chapter 8. We're going to keep moving through chapter 8. So we had our little introductory lecture. Now we're going to move into talking about eukaryotic cells. That's pretty much what we'll be talking about the rest of these lectures. First looking at mitosis and the cell cycle in eukaryotic cells, and then meiosis in eukaryotic cells, and then a little bit at the end about, again, sort of how things get weird. But let's talk about the cell cycle and mitosis. So eukaryotic cells, as we've seen, are generally just more complex than prokaryotic cells, and that includes here at the nucleus level, right? Prokaryotic cells don't even have a nucleus, right? That's enclosed by a membrane. They just have that sort of nucleoid. Because the eukaryotic cells have more complex nuclei, they have a lot more genes than prokaryotic cells, they have a lot more chromosomes than prokaryotic cells, and they're all encased in that little nuclear envelope. The process for duplicating and dividing eukaryotic cells is a bit more complicated. Now, in each chromosome, you'll find essentially a single long DNA molecule. I think it stretches like from here to the moon 10 times or some crazy thing like that. But most of the time, DNA exists in what we call the chromatin format, which is sort of this kind of fibrous, kind of stringy, if you think of like corn on the cob, little hairs on corn on the cob. It's sort of like that inside the nucleus. And generally you don't see that with a light microscope. But if you have a cell that is going through its division processes, then the DNA kind of gets all coiled up and packed real tightly together, and then you can see the DNA molecule inside the nucleus of the cell. So before a cell does start to divide, just as we saw in the prokaryotic cells, the chromosomes will duplicate. Here in eukaryotic cells, they're now called sister chromatids, which means they have the same identical DNA, the same identical traits, and essentially they get kind of connected to each other. The division process involves separating those two sister chromatids, resulting in two daughter cells that are genetically identical to the parent cell and to each other, each containing a complete and whole identical set of chromosomes. And that's what you see here. So here we have our chromosome with the DNA. The chromosome duplicates. We now have the two sister chromatids with the two identical DNA. The sister chromatids separate. We have two daughter cells. And you can see this is what it actually looks like microscopically using an electron microscope and a very, very high magnification. It's all just very tightly coiled DNA there, which is pretty cool. So sort of like people have a life cycle, right? Embryo, fetus, newborn, infant, baby, childhood, adulthood, senescence, old age. Cells also have a cycle of kind of growth and division and so on and so forth. I sort of like to think of interphase, which is when a cell is sort of doing its thing, kind of like childhood, adulthood, and then old age. And then in the mitotic phase, that's our actually division phase, where we will split not only the nucleus, but also the cell itself. So interphase begins with the G1 phase, which is sort of a growth phase. The cell makes all the proteins that it needs to make to do its job, and so on and so forth. During the S phase, S stands for synthesis. This is when the cell is kind of doing its job, being a liver cell or a plant cell or whatever kind of cell it is. But it also includes replicating the DNA in this phase. And then there's sort of a second G2 phase, where the cell is sort of getting ready to go into its mitotic phase, its division phase. So you can kind of think of the G1 phase as childhood, the S phase as sort of adulthood, and the G2 phase as sort of old age for the cell. So mitosis is the distribution of our duplicated chromosomes, they got duplicated during that S phase, into 2 daughter nuclei. Now, one thing I want to mention here, when we talk about daughter cells, we're not talking about gender or sex, right? We're not talking about girls and boys. We're only talking about, it's a sort of gender neutral term, even though we're saying the word daughter. We're just using that as sort of the resultant offspring of these cellular division processes. So during mitosis, the chromosomes get all condensed and coiled up into each other. We get the formation of a mitotic spindle, which is this very specific set of microtubules that move the chromosomes initially to the middle of the cell. The sister chromatids then separate and are moved to the opposite poles, opposite sides of the cell. The 2 new nuclei will form and the cell will split. So there are sort of multiple phases to mitosis, and we start with prophase. So here you can see our cell in interphase. You can see again, you really can't see the DNA very well with the light microscope because it's in this chromatin format. A cell in late interphase will have already replicated its DNA. So in this chromatin is 2 sets of DNA. In the prophase, you'll see the DNA start to condense and coil into the actual sister chromatids, connected at something called a centromere. You get the formation of the mitotic spindle, microtubules from these centrosomes. In what we call prometaphase, you'll see the nuclear envelope sort of starts to fragment. The sister chromatids will attach at what are called kinetocores. Kineto means movement to the microtubules. The chromosomes are lined up during metaphase along this sort of imaginary plane halfway across the cell, our metaphase plate. The sister chromatids then separate into, so now we have full-fledged chromosomes. They're sort of walked to the poles of the cell as the cell sort of elongates during anaphase. And then the last phase of mitosis is telophase, where we get our nuclear envelope sort of forming again. The chromosomes start to uncoil. Come back for a second. The microtubule, mitotic spindle apparatus disintegrates and just goes back to being the centrosomes. And we get the start of cytokinesis. So cytokinesis is the division of the cytoplasm, sort of overlapping there with the end phase of telophase. But it happens differently in plants versus animals. In animals, we call a cleavage furrow. In plants, we get the formation of a cell plate. So the cleavage furrow in animals is sort of like a drawstring. It's essentially sort of a ring of proteins that pinches around the middle of the cell, pulls it really tight, and the cells just sort of pinch off into the 2 daughter cells. Conversely, in plant cells, because they have those cellulose cell walls, cytokinesis occurs by the formation of this cell plate. And so you get the vesicles containing the cellulose essentially starting appearing in the middle of the cell. They merge into the cell plate. The cell plate then elongates and essentially becomes the new cell wall for both of for each of those 2 genetically identical daughter cells. So whether we are talking about plant cells or animal cells, keep in mind then that the daughter cells are genetically identical to each other and to the parent cell. Typically, there are sort of density factors related to the way mitosis occurs. If you've ever sort of like looked at your like if you've ever had a cut or anything and you've sort of watched a cut kind of heal itself, it sort of kind of occurs from the edges and then they kind of meet in the middle. And that's sort of what happens if you were to look at a cell in a culture as well, right? So they really only divide when they're attached to some sort of surface structure, and then they'll essentially continue dividing until they touch one another. So it's sort of density dependent. If there's already like a ton of cells there, then you're not going to get a lot of growth there. The other thing that affects how much cell division takes place, or at the rate at which it takes place, is a bunch of growth factors, which are usually proteins that essentially stimulate cells to divide. And most animal cells will actually not divide unless they get the proper growth factors. And in fact, some animal cells don't divide at all. For instance, brain cells in humans have very, very, very hard time dividing. It's why traumatic brain injuries or spinal cord injuries can be so damaging, because those cells don't divide very easily. So here you can see we've got a couple of kind of cells in a culture. They're not like touching each other, but they are kind of anchored to the surface, and so they will divide and make copies of themselves until you have a single layer of cells. If you remove a couple of those cells, then again, they will make copies of themselves until they sort of have healed themselves and replaced the ones that you removed. And sometimes when things get really wacky, cancer cells, which have these ridiculous rates of mitosis, sort of end up forming clumps or masses because they go through mitosis so easily. And here you can see some cells suspended in a culture. If you separate those cells, they will not grow unless you add some growth factors. If you give them some growth factors, then the cells will divide. And that's essentially what happens in the real world as well, not just in cultures in a lab. We need growth factors in order for our cells to go through mitosis. And so you can see here is a little growth factor. The growth factors will usually bind onto these little receptor proteins, and then these receptor proteins essentially kind of talk to the sort of control system for the cell. So essentially what happens throughout the life cycle of the cell is there are sort of various checkpoints, right? So there's a G1 checkpoint. Okay, do we have enough of these particular proteins, of these particular substances to keep going? Okay, we do. Okay, then we go into the synthesis phase. Then there is a G2 checkpoint. Okay, do we have everything that we need to go through mitosis? Can we do it? Okay, we good? We can? We keep going. We get the green light. Oh, we don't have a particular growth factor? Oh, we don't have a particular protein already made? Nope. Stop. Red light. And then there's another checkpoint during the mitotic cycle itself that sort of occurs after mitosis, but before cytokinesis. And if the cell meets the checkpoint, then it splits. If the cell does not meet the checkpoint, then even though they have two identical nuclei, the cell remains. This is actually how we get big multinucleate cells. So I mentioned cancer earlier. Cancer cells are cells that just divide sort of excessively. They just are supercharged and they're going to go through mitosis super readily. That can sometimes lead to a growth or a tumor, sort of a mass of cellular tissue. If the tumor is malignant, then it sort of breaks off from where it initially developed, and typically it gets into the blood or the lymph and travels everywhere else in the body. If you have a benign tumor, then it's going to stay contained. Of course, malignant tumors are much more dangerous. And typically, for a long time now, they've been treated with radiation and chemotherapy. It's very targeted radiation, quite typically, because radiation, just like chemotherapy, can interfere not only with the cell division of the cancer cells, which is what you want it to do, but then also cell division with all the other cells as well. And so you do want to try to keep the radiation and the chemo as targeted as possible. There's been some really interesting things happening very, very, very recently with immunotherapies, sort of activating parts of the immune system to go after these sort of out of control cancer cells. But that's very early in that process still. And really, one of the best ways to think about treating a specific cancer is taking into account sort of other factors about that person, whether that's their age, their ethnicity, their socioeconomic status, et cetera, et cetera. And you can see if you, age has a huge kind of confounding factor for, I think this is breast cancer, as does ethnicity, but even social factors, right? Or where you live, right? All of that needs to sort of be taken into account. And that's one of the kind of interesting things about immunotherapies is that because they often sort of supercharge your body's own immune system, they're very sort of tailored and specific to that person. So I have a question actually for you guys. So most cancer research uses sort of observational methods, right? But when we're trying to see if a particular medication works, right? That's what we're doing now with even developing COVID vaccines, right? We use observational studies, right? But why do we not use controlled studies for cancer? Do you think it's ethical to expose somebody to cancer? Probably not, right? So we have to use observational studies because you can't exactly say, here, person, we're trying to test this medicine. Let me give you cancer to see if this medicine works. Doesn't quite work that way, does it?